What I want to bring your attention to is how two products vary from each other when the active ingredient is the same, when they are indicated for the same therapeutic outcomes, when the dosage form and administration protocol is the same, then how a generic formulation is different from its innovator formulation, and how a generic drug is different from something known as a branded generic, which we often hear about. Also, what really is equivalence? Excuse me. Now, this topic of pharmaceutical quality is extremely essential because we want all our products, pharmaceutical products, to be safe and effective. We don't want any untoward anaphylactic reactions. We want the formulation to be stable during its shelf life and also during after it is administered to the patient. And we want predictable outcomes of whatever medicines you are prescribing. If this does not happen, we often come across a lot of news articles and even in the media where our regulatory authorities conduct random checks and have flagged substandard and spurious medications, which are, which raises a concern, major concern of safety. A lot of
Quality
news
tests by
articles
India's
also
drug
keep
regulator.
coming up.
In
I
its
will not
monthly
run this
drug
video
alert,
because everyone must have seen this in the news. And often our patients, your patients, bear the brunt of poor quality in medication. Just last year, our regulatory authorities randomly picked up almost 85,000 samples of drugs from Delhi, and more than 7,700 of these samples turned out to be substandard or spurious. And this cough syrup story, I don't think anybody missed, which was the saddest of all, where children bore the brunt of poor quality in pharmaceutical manufacturing. Now, cough syrups actually are very simple to formulate. These kind of lapses should not have ideally occurred, but sadly it did. It is also said that a lot of chemotherapy medications which are manufactured in India and exported to other countries are of poor quality. Now, therapy areas like cancer, there is a very short window of opportunity because the first line therapy is supposed to be the most effective line of therapy, and we want it to be effective for the patient. But when these drugs are substandard, are or of poor quality or unfit for consumption, then the patient loses this precious window or small window of opportunity. So when something like this does happen, there is poor quality and the patient has to pay the price, who do you think faces the music? Any guesses? Is it the pharma company? Is it the pharmacist? Absolutely, sir. It is the doctor every single time. And the same patient who was so respectful, happy with your treatment, and friendly with you suddenly becomes angry, hostile, and sometimes even abusive. And that is why pharmaceutical quality is a very important aspect to pay attention to, because it is only quality in the drugs that you prescribe is what gives you confidence as the treating physician that this medicine will act as predicted and as claimed. So, coming to a little more detail about pharmaceutical quality, when generics are manufactured, they are usually manufactured after the patent expiry of an innovator molecule, have the same active ingredient, dosage, indication, and expected therapeutic outcomes. Now, the innovator has spent a lot of time and millions in research right from the beginning and has established the safety and efficacy of their innovation through phase 1, 2, 3 clinical trials and have established strongly the safety and efficacy. But when generics are manufactured of any innovator molecule after patent expiry, phase 1 and 2 clinical trials are not mandatory. And in some cases of generic manufacturing, even phase 3 and 4 clinical trials are not mandatory, and also bioequivalence studies are not mandatory in some generic manufacturing. Now, I'm talking about bioequivalence because the absence of bioequivalence means that the generic formulation will behave like the innovator molecule. When there is no bioequivalence, you don't get the same clinical or therapeutic outcomes as the innovator molecule. And these differences can often be spotted during the developmental stage of a formulation, which is very important. And if it is not proven, common issues like solubility, like dissolution, come up when the drug is administered. Now, this happens because either the polymorph is not the most stable or the most purest, the particle sizes or the zeta potential does not match with that of the innovator molecule, and hence there could be variation after administration in the absorption, distribution, and even elimination pharmacokinetics of the generic formulation. Now, it is well established that if bioequivalence is not proven and a generic formulation is not bioequivalent to the innovator, the clinical and therapeutic outcomes are not same as the innovator molecule. And there is enough data like this one article which showed that the plasma levels of phenytoin were 31% lower when the branded drug was replaced by a generic formulation. Same happens with tapentadol or any opioid analgesics for that matter. Now, in opioid analgesics also, it's very important that the patient gets the right therapeutic dose. If it is a substandard or a spurious quality medication, then either the patient will require a higher dose of the opioid leading to dependence, or it may cause more adverse effects. So it is very important that these drugs which are manufactured are therapeutically equivalent to the innovator molecule. Even FDA says that pharmaceutical equivalence is when the active ingredient is same, the dosage, the expected clinical outcomes are the same. But this is pharmaceutical equivalence, but only when you pair it with bioequivalence. It's a test which proves that the pharmacokinetic and dynamic parameters of the innovator is the same as of the generic formulation is the same as the innovator, is when you get therapeutic equivalence, which is of major significance to you as the treating physician. So, while most generics do not have to conduct phase 3, 4 and bioequivalent studies, it is the bioequivalent branded generics in India which not only carry out phase 3 and phase 4 clinical trials but also carry out bioequivalent studies, go one step beyond the innovator and now establish the safety and efficacy of their drugs with the innovator in the Indian population now. So the real comparison of any innovator molecule when shifting therapy from innovator to generics is with a bioequivalent generic. Now, this is of particular significance today and the objective of today's session, because you as treating physicians are faced with multiple options of the same molecule. And information like this helps you take the right informed decision on which brands to select for your patients. So when we speak of pharmaceutical quality at MSN, it's not only the API that we focus on. The active pharmaceutical ingredient, of course, is the main ingredient, which is expected to give the desired therapeutic effect. But there is a lot more that goes into making this medication. There is a raw materials and excipients. Today, the market is flooded with various options of excipients varying from 800 to 80,000 rupees per kilo. So, what is the quality of excipient that was used in manufacturing? Which type of polymorph was used? Then every processing facility and equipment is different. Were these maintained to regulatory standards? Because when the API is being manufactured in these processing facilities and machinery, at every chemical reaction, an impurity is left behind which needs to be removed, which needs to be monitored and removed. And after all this comes the color, the coating, the packaging, the storage facilities, all of which impact the quality and stability of a formulation. At MSN Labs, this company ranks number one globally in the number of drug master files filed in the US as compared to any Indian company. We have 25 manufacturing facilities in India and one in the US at New Jersey. And all of these facilities are certified by more than 30 regulatory authorities from across the world, including FDA, TGA, MHRA, and even the WHO. And these certifications are renewed in a periodic manner. So when we carry out equivalence of our formulation with the innovator molecule, we carry out pharmacokinetic and dynamic studies, clinical endpoint studies, and also dissolution studies. But before I move on to dissolution study, I would like to mention here that when we say we carry out bioequivalence in our R&D facility, it is not just any test that we carry out. Bioequivalence studies are completely regulated by the drug regulatory authorities and the CDSCO right from the time the protocol is made. We also have to conduct these studies in CDSCO approved facilities only. And once this is done and the report is made, it is very thoroughly scrutinized by the SEC committee. We are grilled, asked questions, asked for clarity, and only once they certify is when we can claim that our formulation is bioequivalent to the innovator. Dissolution study comes much before the launch, wherein we conduct or we mimic gastrointestinal environment in the lab, and we take multiple readings of the innovator drug and our formulation, and we compare to see that the dissolution is similar. So at MSN here in Hyderabad, actually we have one of Asia's largest R&D center and manufacturing facility, through which now this number is a little old. We have rolled out more than 500 bioequivalent formulations across 50 scientific labs with more than 2,200 scientists working round the clock. And we have more than 1,000 patents for APIs and formulations to our name. With that kind of background, towards the last few slides of my presentation, I want to run you through quickly the bioequivalence that we have carried out with most of our formulations, especially in the diabetes range. And most of these drugs are also first time in India innovations. So we carried out bioequivalence for our SGLT2 inhibitor empagliflozin, which is EPA1, and have established its bioequivalence with the innovator molecule Jardiance of Boehringer Ingelheim. Similarly, we have established bioequivalence with dapagliflozin or our brand of dapagliflozin DAPA1 and established bioequivalence with Vaxiga by AstraZeneca. And our unique or novel proton pump inhibitor, which is very commonly used by diabetologists as well, is a dual release proton pump inhibitor, which releases the drug in two phases from the same capsule and gives a truly once daily performance with a longer AUC, and have established its bioequivalence with the innovator Dexilent by Takeda. Similarly, we have launched first time in India branded bioequivalent generics across various specialties, including the therapy area for pulmonary artery hypertension, heart failure, diabetes, urology. We have almost the entire range, neuropsychiatry, gastroenterology, and pain management, gynecology, and many other super specialties. So yes, we are a world-class integrated global Indian company. We have our own R&D, API, and formulation divisions, and we roll out one product at a time for all batches. As in, we don't have a separate batch for India and separate for export. The same quality medication is marketed and in India and also exported to more than 100 other countries. This is our R&D facility at Hyderabad actually, and I would like to invite all of you to give us a couple of hours and take you to the facility so you can witness for yourself the high quality standards, the certifications, the quality parameters that are followed here. Thank you so much.